this point, we have been talking about Mendelian genetics. We've been using Punnett squares to show a monohybrid cross, something where it's an easy trait to follow, one trait coming from mom, one trait coming from dad. When we put them together, we have an offspring, and it's only been one trait. Typically, also, these have been following the dominant versus recessive rules. So if we have a dominant trait, then that is going to be expressed. If there's a recessive trait along with the dominant, only the dominant gets expressed. And if there's two recessive traits, then that recessive trait is expressed. Not all traits follow that simple rule, however. So this is going to talk a little bit about some of those complex patterns of heredity. So we're going to talk about incomplete dominance, codominance, and some of the other actual things that happen in reality. So some of the things that we've already talked about, like eye color, blood type, hair texture, these actually don't follow the simple dominant versus recessive rules that we've been studying in class. So let's take a look at some of these. The first one we're going to take a look at is something called incomplete dominance. And incomplete dominance is something where we have two dominant traits that can be expressed. Neither one of them actually shows complete dominance over the other. So for instance, in incomplete dominance, you typically have a heterozygous phenotype, which is intermediate between two homozygous phenotypes. And this is because neither allele is completely dominant over the other. Hair texture is a very good example of this. So in class, we've been talking about hair texture as if only one trait and one gene actually codes for this, but in reality, it doesn't. So for instance, if somebody, let's say a mom has straight hair and a dad has curly hair, what might happen is that when they have an offspring, their child might have wavy hair. And this is because hair texture is typically something that's incomplete dominant, meaning that neither dominant traits are actually fully dominant over the other. So you can kind of have a mixture between the two. So in this case, straight or curly, you might have wavy hair. Another type of funky little thing that happens is something called codominance. And codominance is actually where you have a heterozygous individual that expresses both alleles because they are both dominant. So for example, we have feather colors in chickens. So we have black feathers and we have white feathers in chickens. So you can have a chicken that might have black feathers. You might have a chicken that has white feathers. Let's say a black feathered chicken mates with a white colored chicken and they have babies. Well, neither one of these is actually dominant over the other. They're actually both dominant. So what happens here is that we actually have a dominant, or two alleles are dominant, and it shows both traits. So in this case, in the picture, we have the chicken that has both black and white feathers. So neither one of them is actually fully dominant. They're both dominant. And so there's no mixing like we have in incomplete dominance. Each one is fully expressed in the individual. We also could have something called multiple alleles, and this actually happens more often than not. So multiple alleles are basically genes that have 3 or more alleles that code for a specific trait. So typically we've been talking about 2, black and white, purple and white, tall and short, blue eyes, brown eyes. However, just your human blood type is controlled actually by 3 different alleles. So we have here is the fancy way that our blood is written if we were to do these the fancy way. So I is typically the word or the letter that we use when we're talking about blood type. So there's 3 alleles that code for our blood type. We have A, we have B, and we have O. So for example, We also know that we have 4 blood types. So these 3 alleles will code for 4 blood types. We have blood type A, blood type B, blood type AB, and we have blood type O. So blood type A and blood type B, if you notice from the top, are written a little bit differently. We have an uppercase I with almost like a superscript of A for type A blood. And then we have an uppercase I with a superscript for B blood. Now O is coded as the recessive trait. So we use the lowercase I here. Now have a note here that says that both the A allele and the B allele are codominant. Neither one is going to be dominant over the other. However, little I, is recessive. So our blood type O is recessive. So if we take a look here at how we might write the genotypes. Blood type A genotype is typically written as uppercase I, superscript A, uppercase I, superscript A, or it could be written uppercase I, superscript A, little I. If we're doing this formally, this is actually how it would be written. However, if you prefer, and it might be easier if we ever do any of these in class, that we write our the genotypes like this, just big A, big A, big A, big O. And the reason why we do this is because it's a little less formal and it might be a little bit less confusing. So our genotypes, remember for blood type A, could be that the mom and the dad both give an uppercase A allele, or it could also give one of the parents give an uppercase A and a lowercase I. So in this case, or type O. In this case, that's why blood type A has 2 different genotypes. Same with blood type B. It could be written as big B, big B, big B, O. Blood type AB, that's codominant. There's only one way to code for that, and that is big A, big O. Now, if we were blood type O, like myself, formally we would write that as little I. That is the recessive trait, always. If there's an A or a B present, it's always recessive. Blood type O, informally, we can just write big O, big O. This is something we'll be practicing in class. So if you're a little confused at the moment, just hang tight, write down some questions, and we'll get to them in class. Now comes one of the funner ones, and this is something called polygenic inheritance. Now this is where we have a trait that's controlled by more than one gene. Up until this point, all the traits that we've been talking about really are controlled just by one gene. However, polygenic inheritance means that there's these traits that are controlled by more than one gene. Human skin color is an example of this. You've noticed out in society, we have skin colors that range from very pale to very dark. And that's because there's such an array of combinations that we can have for skin color. Now, there's 3 that are just written here on the slide. We'll show you a little graph here in a second. So somebody that has very dark skin might have the dominant alleles for all of those genes that are coded, they're used to code for human skin color. So for example, dominant alleles for genes A, B, C are basically produce melanin, which is the pigment of the skin. So the darker the skin, the more melanin. So very dark skin type might have basically dominant alleles for all 3 of those. Whereas somebody that has very light skin might be recessive for all 3 of those genes. We can also see an array. So you could be dominant for A, maybe lowercase, or be recessive for a B, and be heterozygous for a C. So there's a range of possible skin colors here. Now, polygenic inheritance also deals with height, eye color, and weight. So I know in class, sometimes we do our Punnett squares with blue eyes, brown eyes. But unfortunately, it's not as easy as we've been doing it in class. Essentially, it's all about polygenic inheritance. These are traits controlled by more than one gene. So this is just an example of a possibility of skin colors that we could get from very dark to very light in humans. So if we, again, just using the ABC genes, notice that our ABC up here is heterozygous for each of these. If we cross that with a person who is heterozygous also for all three genes, there's a possibility of all of these particular combinations. So it's not as easy as two light-skinned people having a light-skinned baby. It doesn't always happen that way. It's always because the genes that code for it, there's multiple. So it could be an array of genes. We have no idea what the combination is going to be. We also have something called pleiotropy. Now, these are where genes are influenced by multiple characteristics. Now, typically we have, let's say in this example we have here, we have the shape of our blood vessels, our blood cells, our red blood cells specifically. Now, sometimes our red blood cells can have a sickle shape. And the sickle shape that you see here in this picture is actually a disease called sickle cell anemia. And what happens here is that the red blood cells tend to get this long, elongated shape that looks like a sickle. And these get stuck in the blood vessels. They'll clump up together. They cannot move easily through the blood vessels. They'll start to clot, and that causes a lot of problems. So this could possibly cause heart failure, could cause brain damage, could cause weakness, anemia, etc. So it doesn't just cause sickle cell anemia. It can actually have a large array of characteristics. So for example, if we're looking at these red blood cells and we have a dominant, which is big R. Big R is the dominant for round-shaped cells. Now you also could be heterozygous as well, so an uppercase R, lowercase r. Now if we have that lowercase r, that means that there's a recessive trait for a sickle shape. And if we get two lowercase r's, so for homozygous recessive, then that could cause some of those cells to become sickle or have that sickle shape. Sometimes we also see traits determined by our sex because we have some genes that are carried on our sex chromosomes, typically the X chromosome. Now, remember, females, we have two X's. Males, you have an X and a Y chromosome. This is what determines our sex. Now, sometimes we have these traits, and they're typically carried on the X chromosome. Now, for example, one of the biggest ones that we know of that's sex-linked is color blindness. Because men only carry one X chromosome, sometimes we see that trait expressed more because there's a possibility that there's that dominant trait or that other trait that's not able to not express it or be expressed. So color blindness is one of those biggest ones. So females, for instance, let's take a look here. So females carrying along the sex-linked gene of color blindness, if we have a big N, that's basically a normal person. This person happens to be female because there's two X's. Now notice that superscript up here, that superscript is N, that's the trait. It's being carried on the X. It's very important that that X shows up if we were to do these in class, in problems, that X must show up. Now a female can also be a carrier. They're typically normal, so they see color as normal, but that means they might be a carrier. So notice that we have an uppercase N, which is the normal, but we have a lowercase n as well. That means they are a carrier of color blindness. We also have possibility to see females that are carriers, that are actually colorblind, and that's because they have two little n's, so they actually show the recessive trait to be colorblind. Now, this can only happen if the female has 2 lowercase n's. Now, a male, however, only has 1 X chromosome, so they only have a 50-50 shot at being normal or being colorblind. So if it's carried on the X and they have the big N, that means they're normal. They are actually okay. They can see color just fine. But if they're carrying that lowercase n on their X chromosome, that means they might be colorblind. So males for sex-linked genes tend to express some of these a lot more because they only have a 50-50 shot. They only have that 1X. Now, sometimes we have environmental influences on gene expression. And so this really doesn't have any necessarily DNA consequence, but sometimes the environment can influence what gets expressed. So sometimes external environment influences could be things like temperature, could be the light or the amount of light, could be chemicals, could be infectious agents. Some internal environments could be related to hormones or age, nutrition. So some of these things, such as like going gray or silver, some of those different colors that we see, sometimes they're actually influenced more by environment than by gene. And also sometimes we see the color of fur or height and weight. These can also be influenced by environmental influences, not just necessarily our DNA. So an example of this would be an Arctic fox. So here's a picture of the same species of Arctic fox, but they look very different. This one on the left is brown, the one on the right is white. Now, the fox's fur color is actually expressed by an environmental influence of temperature. So if it's really cold out, they have to blend in with their environment somehow. So this way, they turn white because it's a little bit more snowy. It's kind of used as a camouflage. But when it gets warmer, there's no snow on the ground. Their fur becomes brown, and then this way they're able, again, to blend in with their environment. It's an evolution thing. Makes sure that they are camouflaged and protects themselves from predators. Another example could be human height. This is based on nutrition. So even though you have maybe a height gene for being 6'5", but if you don't get the proper nutrition as a child, as a baby, and growing up, you're not going to reach that 6'5" potential. So a lot of times nutrition has a huge influence on human height and weight as well. Leaf size can be influenced by the environmental factor of light. So it depends on if it's a shade leaf, a sun leaf. If it gets a lot of light, it tends to be a little bit smaller and thicker. If it's a shade leaf, it tends to be larger, a little bit softer.